So thanks very much for the kind invitation to come here and for the also very kind introduction. And I'm going to try to talk today very briefly about the aging research landscape that I think is getting very exciting. And what I will do is introduce that at the beginning of my talk, and later on talk a bit more specifically about what we do in our lab, that is senescence, and see how it can apply to aging research. And just to mention that now our institute has been rebranded as the MRC London Institute of Medical Sciences. And in fact, one of the tasks that the MRC has proposed for our new institute is try to study multimorbidities. So hopefully we are not part of UK Spine, but maybe we are involved in the future, and we are collaborating, of course, with some of the groups involved there, and some disclosures of my collaborations or involvement with some companies that are working on senolytics mostly. So just to try to start, and this is something that is, of course, familiar for everyone that comes here. So we scientists usually see everything as a problem that we try to solve. And here the problem that we try to solve is aging, but it's a problem that comes from something that is really nice. And this is that the lifespan, the amount of ages that people live has been expanding almost constantly for almost 2 centuries. And that's obviously really nice. So people 2 centuries ago just lived 30 years. Now they can live up to 70 or 80 years. So that allows to do many more things in life. But of course, that comes to a price, and the price is that the population gets older. And this is in color, what we are seeing is the percentage of the population, in this case of the UK, that is over 65 in 2016, and was the projection in 2039. And we see that it is already very high, especially in some areas, and it will just increase. That again is something that is good. We are extending the life of people, but it comes to a price. And the price, I think that we have introduced already the concept, and this is in fact one of the main focus of this conference, is the difference between lifespan and healthspan. So lifespan is what we live, and 100 years ago we lived for a shorter time, so probably around 50, 60 years as an average. Today we live much longer, but we have this healthspan, that is the amount of time that we live and we are healthy, that has not increased in the same proportion that the lifespan has increased. And we have this gap of ages at the end of our lifetimes where there is like age-related disease. And what we have to try to do is to try to close this gap, and of course keep trying to increase in the lifespan, but try to have this healthy life. This has different consequences, both medical, clinical, and also economical. So the first consequence is that as we get older, the amount of people that has several diseases, several chronic disorders, increases. So if we just look into people that are in the 40s, so there will be just very few that have more than 2 chronic disorders, that are living with more than 2 chronic disorders. So instead, if we look people that are living in the 80s, so that clearly increase, and there will be people living with 3, 4, 5 and more disorders. So that obviously impact in the quality of life, and also impact in the treatment that they have to have. They have to have many pills for these different diseases, and that complicates things and creates problems by itself. Of course, the quality of life and how people feel is the most important, but that also impacts economically. So if we look into what older people cost to the NHS, the cost spiral as people get older. So there is clearly a double incentive, both health-wise and economic-wise, to try to improve the health span. And that's what we aim to do. So rather than just increase what people live, so increase the lifespan, try to close the gap between this lifespan and health span. So try to achieve that people live healthier for a longer time. And that's what aging research is aiming to do. And research into aging has advanced a lot in the last 40, 50 years. So if you think about it, so how people age is probably one problem that you will think that is almost untractable. So it's highly stochastic, there is a lot of variability, and there are many causes involved. And you will think that it's very difficult to try to make some sense, to try to research into it and try to get some underlying causes. But as I mentioned, as research advanced in the last 40, 50 years with the use of model organisms, with the advance of biochemistry, with the advance of genetics, we are having a much better idea of how we age and what is behind this aging process. And what people has tried to categorize is that there are a number of hallmarks or a number of processes that are involved in this aging process. And these are different hallmarks that might be biochemical, genetic, or cellular, and that correlate and have some relation and causality with aging. And I'm not going to mention all of them, but for example, I'm going to just pick a few of them, and I will go through some of the others. So what researchers have realized is that, for example, telomeres, the very end of the chromosomes, get shorter with age, and that causes a number of issues that cause DNA damage, and that's involved in signaling and create some of the problems that are related with aging. Cells, for example, undergo this process of cellular senescence that I will talk later on in much more detail. And this accumulation of senescent cells that are basically stress cells that accumulate in the aging organism has a causality and has an involvement in aging. And there are a few other processes, like, for example, exhaustion of the stem cells that we have in the body, or deregulation of nutrient sensing. So I'm just going to show an example of one of these processes and how it goes wrong with aging. And I'm just going to concentrate in one of them, that is loss of proteostasis. And I think that's what I'm going to talk about. This will probably will apply in general terms to the other processes. So the proteins, so for the cells to work, the proteins have to be properly folded. But of course, there are like different stresses, both internal and external, that impact into that and make that proteins don't become folded properly. So we have these unfolded proteins. And this is in response to different stresses. And these stresses, for example, happen normally, but will accumulate during aging. And when we have a problem, so the cell usually has different solutions for it. And this again can be generalized. So this unfolded protein, so the first thing that the cell will try to do is try to see whether it can solve it. So we try to see whether there's a way in which it can refold the proteins, and for that involve chaperones. And if it sees that the damage that it has been done in the proteins is too much and it cannot cope and repair it, in this case, refold the proteins, what it will do is try to, in this case, degrade it. So degrade the proteins by using different systems, either proteasomal degradation or autophagy. And usually the cell will try to cope with different kinds of damage in this way. So you will have some damage that will happen normally during your life, but will accumulate more during aging or in some diseases. And the cell will have like mechanisms to try to sort this damage. And when it cannot sort the damage, it will try to basically eliminate the cause of this damage. And the same applied to, for example, to DNA damage, where the cell will try to repair it, and if not, will try to eliminate these cells that have DNA damage. But what happens, especially during aging and more, is that some of these mechanisms will be saturated or will not be working normally, and the cell will not be able to refold all these unfold proteins, or will not be able to degrade all of those because these systems will be dysfunctional or will be saturated. And in those cases, you will have an accumulation of a sort of damage. In this case, will be these aggregate proteins that will contribute to aging. And so this is, for example, what happened during proteostasis, but DNA damage or even accumulation of senescent cells will follow a similar pattern in which the cell will try to deal with this damage. We'll have different mechanisms to try to do that. And when those fail, there will be an accumulation of this damage of dysfunctional cells that will contribute to aging. So the other side of the coin of understanding some of these basic mechanisms that are underlying the aging process is that potentially we can target some of these underlying mechanisms to try to treat some of the age-related diseases and try to improve the health span. And this is just a summary showing that many of the hallmarks of aging that I mentioned before can be targeted in one another way. And I will talk in the next slide a bit more in detail about some of these. Some we have already strategies in which we can target that are in progress. Some have been tested or they are in clinical trials for some processes. Others are more theoretical, but has the potential to be targeted. For example, these are like some of the intervention strategies that we can use to target some of the hallmarks of aging and improve the aging process. One of them is targeting factors in the blood that contribute to aging, and this has been basically shown through a number of very basic research experiments, what we call parabiosis, in which we have like old mice connected with young mice, and then what it was shown is that if all mice have the blood of young mice, you will improve many age-related phenotypes. And eventually what this has taken is to try to discover some of the specific factors, and this is an area in progress, blood factors that if you just give some of these blood factors to an old, in this case, an old mice, you can improve some of the age-related phenotypes. So I think that this is an area in progress. So there are some targets like TIM2 that have been discovered, and there are still many things that has to be done, but has the potential to improve age-related diseases and phenotypes just by giving some blood factors that mimic what happens in the blood of young animals. One of the potentially strategies that has been studied more in detail is try to target metabolism. And for many years, for example, has been known that if you just have a limited diet, a caloric restriction, mice and different organisms can live longer. And we know already some of the underlying biochemical pathways. For example, the mTOR pathway is involved in controlling this sensing of nutrients. And there are inhibitors, or there are drugs that can mimic some of this manipulation of the metabolism. And there are accumulation of data, and I think that we'll see even some data during this conference that using mTOR inhibitors, such as rapamycin, that is used in the clinic for immunosuppression. By using these mTOR inhibitors, you can extend the lifespan and the health span of a cohort of model organisms, and potentially this could also work in mouse, and this is in humans. And this is one of the strategies that probably has been studied for a longer time, and there is evidence for this manipulation of nutrient sensing from yeast to mouse and even to bigger mammals. Other of the strategies that is right now very much in fashion is cellular reprogramming. So this is a field that started probably like 10, 15 years ago. So the main discovery over there is that you could have a somatic cell, and you could reprogram it into a stem cell by expressing a number of reprogramming factors they are called Yamanaka factors. But one of the things that was discovered is that if you do that in vivo, there are like two processes. So this is a two-step process. So eventually you could reprogram these somatic cells into stem cells, and potentially that's something that you don't want to do in vivo, but you go through an intermediate process where there is some de-differentiation. And this partial reprogramming is a strategy that has been shown to improve many of the age-related phenotypes. And in fact, I don't know if you have been following the press, but there is like a company that is called Altos Labs that has been funded by Bezos and many others, so the founder of Amazon, that is investing in this strategy to try to target age-related diseases. And finally, one of the other strategies other potential interventions that can happen that relate to these hallmarks of aging is targeting senescent cells. And I will talk about this a bit more in detail, and I will go through some of the examples because this is the specific area of research in my lab. So this targeting of senescent cells we refer as senolysis and the drugs that can do that as senolytics. So basically, just to introduce, so cellular senescence is a process that was discovered by serendipity in 1962, so more than 50 years ago, by Leonard Hayflick. So he was just intended to grow viruses, to study the viruses and vaccines. And for that, he cultured primary cells. And by culturing these primary cells, one thing that he realized is that contrary to what was thought at that point, primary cells could just replicate for a limited amount of time. And eventually after that amount of time, they will have this phenotype that he calls cellular senescence, that is just a cell-related phenotype, but that he thought that might mimic what happened during organismal aging. But when we refer to this cellular senescence, we refer to this cellular phenotype in which cells enter into this state of replicative exhaustion. With the pass of the years in 1997, so what we realize is that a state of cellular senescence not only happens in response to replicative exhaustion, but also when the cells are exposed to different stresses. And one stress, for example, is the expression of oncogenes. And the cells, when they express oncogenes, they go into this state that is called oncogene-induced senescence. And they do that to try to avoid to become cancerous and transformed. But basically, it's a phenotype that is very similar to this senescence that happened during replicative exhaustion. And what we realized a few years later in 2005 is that senescence is not only something that we scientists can see in the lab and that we can just reproduce in cell culture, but it's a phenotype that happens in vivo. So in 2005, what a number of researchers realized is that this oncogene-induced senescence happen in premalignant lesions, so in tumors that are not malignant, and that by inducing this senescent response, you avoid progressing to a more malignant tumor. So senescence basically happens in vivo and can limit the progression to a more malignant state. And perhaps the big breakthrough that happened with senescence was like more than 10 years ago, so in 2011, that a number of papers from the Jan van Dersen lab show that if you specifically kill senescent cells in a mice, the mice can live longer, and not only live longer, but also live healthier for a longer time. So you extend both the lifespan and increase the health span of these mice. And you can target a number of different diseases. And this makes the scientists excited on trying to find drugs that could mimic this extension. So just before we go into that, very quickly, again, I just want to mention that when we talk about senescence, is what we are referring is to a cell phenotype that is activated in response to different stresses and that induce many changes in the cell. So for example, the cells become arrested. So they are arrested in the cell cycle and they don't proliferate anymore. They change the shape and the morphology. They start to produce different secreted factors, including inflammatory factors that can call the immune system, and they become more resistant to apoptosis. And this senescence is a growth arrest that is different, for example, from the growth arrest that happened during differentiation or during quiescence. And some of the things that's differentiated is that is highly stable. So the cells, when they enter into senescence, they cannot come back or cannot come back so easily, and also happens in response to damage. And on top of that, there are a number of phenotypic characteristics, including this inflammatory phenotype, this expression of inflammatory factors that are associated with senescence. So at the molecular level, the growth arrest is controlled by two of the main pathways that control cell cycle and that are often also mutated in cancer. One is the p53 pathway that activates a cyclin-dependent kinase inhibitor that is called p21. And the other is this p16 RB pathway. And what they do is, in response to these senescence inducers, activate these pathways to basically exit cell cycle and arrest the cell. So this is what controls the senescence growth arrest. And on top of that, as I mentioned, one of the main characteristics of senescent cells is that they express different secreted factors. These are inflammatory factors, but very often what they are there is for calling the immune system and interact with other cells. And just at the molecular level, this is controlled by different pathways, like for example, the SIGAS-STEAM pathway that sends different kind of damage and activates eventually transcription factors such as NF-kappa B that controls the expression of some of these cytokines that are secreted by senescent cells. So as I mentioned, senescence, on one hand, senescent cells accumulate during aging. On the other hand, they are present in premalignant lesions and protect against these premalignant lesions. And there is a paradoxical role for senescence that I will just very briefly talk about. But I want just to very quickly mention some of these points. So senescent cells, for example, are present in cancer and fibrosis, and they limit this progression to cancer or the severity of fibrosis. Senescent cells also accumulate with age, and they are associated with multiple diseases. This is a not exhaustive list, but you have senescent cells present and contributing, for example, to osteoarthritis, to COPD or IPF, to diabetes, and to, of course, cancer and fibrosis, and many other diseases, including neurodegenerative disease. So there is a paradoxical role of senescence. On one hand, senescent cells accumulate with age, on the other, protect against cancer. So for many years, there was this situation in which we have this balance, in which we didn't know, researchers, we didn't know what we had to do to try to use senescence to our benefit. If we were trying to protect against cancer, you would say, well, maybe you there had to potentiate senescence, and then you have a strong anti-cancer response, but that could be at the expense of having increased or premature aging. And the other way around, if you decrease senescent cells to try to decrease the signs of aging, could be that you were being more prone against cancer. And there was these paradoxical roles and this situation that make advancing in how to use senescence for therapeutic use a bit difficult. But the big breakthrough came in 2011. So this is a number of papers from the Jan van Dersen lab. And what they show is they generated a mice, so a totally artificial mouse system that I'm not going to enter into the details, but basically what they have is controlling under the p16 promoter a gene that is a gene that could kill cells when you add a molecule. But at the end, this is a trick to have a mouse model in which you can kill senescent cells at will. When you administer a drug to the mice, you can specifically kill the senescent cells. And what they saw, and the question was, what happens when you do that? Because senescence has these positive roles in preventing cancer, these negative roles during aging and in many age-related diseases. And the big surprise and the big breakthrough is that when you, rather than potentiate or decrease senescence, when you kill senescent cells, what you achieve is that this elimination of senescent cells extends the lifespan of the mice, and the mice not only live longer, but live healthier for a longer time. So these are like almost 2-year-old mice. One has not been treated with this drug that kills senescent cells, the other yes, and you can see that the one that has been treated look healthier. And not only look healthier, but by ablating senescent cells, you delay cancer-related deaths, and this accounts for a big chunk of this extension of the lifespan. But you not only delay these cancer-related deaths, but later on, different studies have shown that by ablating senescent cells, you have positive impact in more than 20 different age-related pathologies. And one of the good things is that you don't need to be treating with a drug that eliminates senescent cells all the time. Intermittent elimination of senescent cells works, and there are not many side effects, and always there are caveats of that. But in the original study, the only side effect that they saw is that if the mice have some wounds, so this will take longer to heal. So the attraction of senescence as therapeutic target, as I mentioned, is that by targeting senescence, we are targeting a root cause served by many diseases of aging. So we are not specifically targeting a disease and having to treat 4 or 5 different diseases with different drugs, but we can target an underlying cause of aging. Intermittent treatment is sufficient to produce benefits, so you don't have to be given drugs or potentially drugs all the time, so you can reduce potentially side effects. And in fact, already there was not very important side effects. And on top of that, senolytic treatments, as we mentioned, they can be used to treat multimorbidities and have the potential to make us live healthier and for a longer time. So just in general terms, this is what's a model that integrates the role of senescence in health and aging, these positive and negative roles. So when you have a tissue in which you have damage, that can trigger senescence. So and this is a response that the cell has, so that the cells that accumulate damage, don't replicate, and this damage is perpetuated in the body. And this cell basically got to this growth arrest, one that you trigger senescence, so it will not replicate. And they will start to secrete these SASP, so these secreted factors that will recruit the immune system. And in this context, the immune system will clear the senescent cells. So you have basically an intrinsic way to arrest the cells that are damaged, and an extrinsic way to call the immune system and eliminate these damaged cells. And in normal circumstances, this tissue will regenerate, and senescence is beneficial. Senescence limits cancer, limits fibrosis, is necessary for proper wound healing, and contributes to development. So the problem comes with when this process does not go like here, and you abnormally accumulate senescent cells. And this can be because you have a chronic cause of damage, or because these senescent cells are not eliminated efficiently because the immune system, for example, when you are old, is not so efficient in eliminating these senescent cells, or even because senescent cells have active mechanisms to try to evade the immune system. And when you accumulate these senescent cells is when you have negative effects associated with senescence. And basically senescent cells that contribute to tissue dysfunction through producing chronic inflammation that contribute to disease, even paradoxically contribute to advanced cancer. And this accumulation of senescent cells, we can reset using drugs that kill senescent cells. That what we will do is basically the same that happened normally. So basically we'll clear these senescent cells, and then these senolytic therapies will have these positive effects that we mentioned before. And these senolytic therapies, so these drugs that kill senescent cells, are the ones that are one of the more popular ways to try to target senescence, but we can target senescence in other ways. For example, by targeting this inflammatory process that is involved with these negative consequences of senescence, or by even by potentiating the immune-mediated elimination of senescent cells. So basically, these are some of the ways in which we can target senescence: killing senescent cells, potentiating the killing by the immune system, inhibiting this inflammatory phenotype, or even preventing senescence induction. The most popular, and I will talk about this, is this senolytics, so this killing of senescent cells. But I'm going to very briefly touch into some research on my lab in which we follow strategies to try to inhibit these pro-inflammatory cells or preventing senescence induction. I will start by this. So for example, one of the things that we do in my lab is different genetic and drug screenings to try to identify regulators of senescence and senescence-associated phenotypes. So in particular here, what I'm going to show you is two screenings that we did using two models of senescence, one of oncogene-induced senescence and another of telomere uncapping. And what we try to identify here is drugs that when we use them alleviate the senescence response. And basically these are the details of the screening, but basically we are the drugs at the same time that we induce senescence. So when when you induce senescence, senescent cells arrest, and we can monitor that because they don't incorporate BRDU and they are not green here. If we prevent that, for example, with inhibitors of TGF-beta, we see that there are more self-replicating because we have alleviated the senescent response. So we screen libraries of drugs, and we try to look for the ones, and we did several retesting, and we try to look for the ones that seems to alleviate senescence in the two different models of senescence. And we identify several drugs, one that we call 3DA, that is the one that we have worked more. And for example, I'm just gonna show you some data. We use this drug 3DA in a model of basically geriatric muscle stem cells. So that's potentially something that is related with sarcopenia and some muscle diseases associated with aging. And what we knew, collaborating with the group of Pura Muñoz, is that induction of senescence in very old mice limits the ability of stem cells to regenerate the muscles. And what we did is try to see whether if we treat muscle stem cells with this drug that we have identified that inhibits senescence, 3DA, we could try to alleviate this process. And we did this treatment ex vivo previous to the transplantation. And what we can see here in green is the muscle fibers after transplantation. If you transplant the stem cells of young mice, you see you have like these thick muscle fibers, and there are many of them, many Greek ones. If you transplant the stem cells of all mice, so you have very few of these that have regenerated. But if we treat these all mice, these all fibers with this drug that alleviates senescence, we can try to reset this process. And we can, of course, monitor that. But by using some of these drugs, we can improve some of the phenotypes that are associated with age, in this case, in a muscle model. So the second thing that I'm going to just show you very quickly is regulation of the SASP, this inflammatory phenotype associated with senescence. So again, using, in this case, the model of oncogene-induced senescence. What we did here is a screen for genes, so using RNAi, that control the expression of these inflammatory factors that can cause chronic inflammation and can be involved in some of the detrimental effects of senescence. So we screen something like 7,000 different genes. We identify a few that can inhibit the SASP. And amongst them, there was a new target that is a splicing protein that when we knock them down, we can basically prevent the expression of these inflammatory factors that are involved in chronic inflammation. And in this case, we use that in a model of, and we can do that without affecting the senescence growth arrest. And what we can see in this case in a model of cancer is that if we inhibit the inflammation, but we preserve this senescence growth arrest, we have some of the benefits of senescence preventing cancer without having some of the associated detrimental effects of this chronic inflammation that fuels cancer in more advanced phenotypes. So by targeting the SASP with these anti-inflammatory phenotypes, we can achieve some of these processes. And perhaps the most popular strategy to try to target senescence are senolytics, as I mentioned, so drugs that kill senescent cells. So my lab has been looking for different of these strategies, both using drug libraries and drug screenings, but also using genetics to try to identify new targets that later we can try to follow up and develop drugs. And what I'm gonna, and some of these are in process, some we publish, some we are in the process of publishing. I'm gonna just very briefly talk about one of the drugs that we identified that can kill senescent cells. So this comes again from screenings when we compare normal and senescent cells. And basically, we look to the drugs that specifically kill senescent cells. So when we identify a family of drugs that are called cardiac glycosides, we identify particularly owabine, and in this case, treating with owabine can kill the senescent cells that are in green here, and as you see, there are no green cells over here. And the interesting thing is that cardiac glycosides, such as digoxin and digitoxin, also can kill senescent cells. And this digoxin and digitoxin have been used in the clinic for many, many years to try to treat cardiac arrhythmia. So these are given to patients. And what we discover is that they can kill senescent cells and potentially we can repurpose as some of these drugs. I'm going to go quickly through some of these experiments, but basically treating all mice with owabine, we specifically eliminate senescent cells that have benefits like the mice have like a better physical, the all mice treated with owabine have a better physical, perform better in physical tests. They also have some metabolic, we reset some of the metabolic effects that we see during aging, like for example, there's a decrease of albumin in all mice that it gets resetted when we treat with owabine. And very quickly, what I'm going to show you just to finish the talk is an example of how we can try to start to translate that into the clinic. And I'm going to go into not an age-related disease, but an example on cancer. And on cancer, the idea that we have is that many of the cancer therapies that we use, like chemotherapy or radiotherapy, cause senescence in the tumor. That's in part how these therapies work. But if we come with a second drug, with a senolytic drug that will kill these senescent tumor cells, we could have a stronger effect on the tumor. And this is what we are seeing here with the example of our cardiac glycosides. So these are like cancer cells. We treat with different chemotherapy, and these cells here are arrested, but they are still there. If later on we come with, in this case, owabine, that is a cardiac glycoside that kills the senescent cells, we can eliminate these cells. So we took advantage, and we did in collaboration with Lars Sender and Bernd Pichler in Tübingen University, of a probe that can image senescence by PET. So this is an imaging probe that can image senescence, taking advantage of some of the characteristic of senescence, is that they are beta-gal positive. And what I'm going to show you is data from one patient, and this is data from Lars Sender. So this is a patient with glioblastoma. So this is a cancer in the brain, very aggressive, was subjected to surgery, irradiation, and target therapy. After a couple of months, the cancer reappeared. And Lars, a part of imaging for the cancer, so he used this new probe that can image senescence, and he detected that in the area where the cancer was, there was this strong signal for senescence. So what Lars did is he treated this patient again with the target therapy, but he gave it digoxin at the same dose that you give to cardiac patients as a senolytic agent. After 3 weeks, so the patient had improved, and when it was imaged, so the tumor has decreased in size, and in fact, the senescence signal has decreased. So of course, this is just one patient, and there are no controls, but this is now in a phase 1/2A clinical trial in Tübingen, this senescence imaging. But the idea is that by trying to use some drugs that we can repurpose and some new technologies, biomarkers or imaging technologies that we can monitor senescence, we should be able to try to translate some of these strategies to the clinic. And just to finish, in the case of senescence, by targeted senescence, we can have benefits in age-related pathologies, as I mentioned, in cancer, but more importantly, potentially in the long term, by treating an underlying cause of aging, we could be able to improve multimorbidities without the need to give multiple drugs. And just to finish, so these are some of the people in my lab and the collaborators that contributed and our funders. So thank you very much.
Thank you very much, Jesus.
Thank you.
Really fascinating talk there and insight into the biology of senescence and those really exciting clinical and translational options that you just showed us. It's fantastic. So we've got some time for some questions. So please, yes. We might send them. The microphone's going around.
Thank you. Very interesting presentation. Thank you very much. I'm a retired specialist in public health and a public contributor as well in my retirement. I have 2 or 3 quick sort of comments. I may not quite be questions, but in your initial slides on life expectancy increasing, in the last 3 or 4 years, life expectancy has actually stalled. And for the first time in maybe ever, it starts to drop. I think one of the Marmot reports in the last few years showed that, particularly in women in deprived areas such as the Northeast. And that's even before that was thought to be possibly due to the austerity from the 2008 financial crash. And we don't yet know fully the impact of COVID, long COVID, deaths, morbidity and mental health, particularly in young people. In terms of trying to improve healthy aging, the leading causes of death at the moment for men are ischemic heart disease, and for women is dementia. And it's my opinion that cancer gets disproportionate attention and resources. And in terms of the senescent cells, all the listings were physical disorders, none in the brain, although you did mention, I think, neurodegenerative disorders. And another comment, a few years ago, maybe 10 or 15 years ago, there was quite a lot of interest in something called the poly pill. It made the front cover of the British Medical Journal. It was a pill that had a low dose statin, low dose antihypertensive, and a low dose aspirin. It got a lot of publicity at the time, but I've never heard much about it since. And in terms of dementia, aging research, there's been anecdotal and some research studies suggesting a diet rich in blueberries, walnuts, and a little bit of red wine helps prevent dementia. I didn't know if anybody's interested in picking up an idea to develop a pill that might contain some of those ingredients for dementia aging prevention. Thank you. Sorry, it's a bit of a mixed bag.
No, so I think that you're right in all. So you're completely right in the. Of course, you are the expert, not me, on the life expectancy. And I think in the curves that you have, you also, I didn't mention, but for example, you could also see that around the world wars, there was a drop in the life expectancy, right? And I think it's a bit more depressing that right now we are where we are. That's probably caused by, perhaps, at least by austerity, we have like a decrease. So I think that you mentioned a few points. I have a few things in mind. So one was related with senescence, and I mentioned I pick a few diseases. I didn't mention neurodegenerative disease, but for example, there are studies that show also that elimination of senescent cells can improve Parkinson, Alzheimer's. There are also studies that relate to some mental disorders that potentially might improve when you kill senescence. But I think that this will need further study. And the other thing that I want to pick is, so I think it's tricky. You say cancer is probably overfunded. So I think that I heard some statistics that said that, okay, that if you just eliminate all deaths by cancer, you will just extend lifespan for, I think that is like a couple of years, because basically you will have people dying from coronary disease, right? So I think that that is true, that any of these causes, maybe some are like more funded and more studied than others, but I think that perhaps one of the things that we are advocating with this aging research is that there are some underlying causes that if we target some of these underlying causes, and I talk about senescence because it's my field of research, but probably the same applies to metabolic, to nutrient sensing or the mTOR pathway, to targeting autophagy, to targeting in other ways. So if you target those, potentially you can improve many of these different diseases. So potentially even you might not need this polypill, but you can have something that decreases some of this damage that accumulate and make general improvements.
Thank you. Can I ask, do you think aging is a systemic pre-malignancy?
If I think that aging is a systemic pre-malignancy, so I think that probably goes into definition, right? Or how do you define things? So what I will just, and you can just define it in one way or other. What I will just think, or the way that I think, is that there are some of these types of damages that accumulate gradually during the time that contributes, and some of these are interrelated, contribute to aging, or some of the diseases that at the end cause this gradual deterioration that is aging. I don't know if you, by systemic premalignancy, you refer to related to cancer or just to?
I was just thinking of some of the data you've shown, you know, this pre-malignant state, and it's affecting many different organs.
I think that's seen in that way, for example, this accumulation of senescent cells, and the same applied to some of the other hallmarks. The way that I see is that you have some of this damage that accumulates gradually. Of course, at some point, there is probably a threshold effect, and a bit more of this accumulation causes a bigger effect. And this is basically impacting at the organ level, but also probably inter-organ level. It's impacting different diseases. So I think that you could refer it like that. Yeah.
Thanks for it. Thank you for a lovely talk, Jesus. Don't know if it's on, but maybe you can hear me anyway. I was wondering about the targets that you've been identifying, whether the sort of direction of effect that you'd want to hit. Are most of the senolytic therapies that you're looking at inhibitors, or are some of them agonists? I'm just wondering sort of what you want to do to those targets, if you want to activate them or inhibit them.
Yeah, so I think, so yes, I'm going to just repeat the question just in case, especially for the people that we are streaming. The question was about the different targets that we have identified, especially the senolytic, whether they were what we are trying is to inhibit or to activate. So I think in most of the cases, so what we have been doing is when we are doing target screenings, either RNA or CRISPR, so it's basically inhibition or knockdown or knockout. So in most cases, what we are aiming is to inhibit. But of course, sometimes when we just get some of these targets, maybe they are not druggable per se, and there are ways that we can just try to look into the pathway, and there might be some ways in the pathway in what we are trying is to activate to cause the same effect. Potentially there are others that are, I didn't talk about that today, but that there might be things that we want to direct the immune system towards senescent cells, and there I guess that we could be thinking of either using antibodies or some other kind of cell therapies to eliminate the cells, or use antibodies for that.
Okay, we're running just about on time. Have we got enough time for one more question, I think?
Hi, thank you. Considering the pleiotropic nature of some of the genes and processes involved, hypothetically, at what point do you think the optimal time for intervention would be in humans, considering the targets? And does this vary for different approaches that you've mentioned, the xenomorphics, the senolytics, And also, would a lower dose be effective earlier, or do we need to be focusing at a later stage in aging?
Yeah, so I think that the answer is we don't know, but hopefully we should know soon. So I talk mostly about senescence and senolytics. So senolytics, the concept has just 10 years. So the first drugs that do that, or that they were discovered doing that, have like probably like 4 or 5 years. So maybe a bit more. First clinical trials, probably they're like 3 or 4 years. And if you just think of the number of clinical trials and they're very early phase in specific diseases that you have on senolytics, and some of them are like first in human, very small scale, probably you have like less than 50, even being generous. If you compare that with the scale of trials that you have for cancer, for example, and that maybe goes not necessarily just to the funding, but also how young this field is, I think that the answer to that is that probably there are different strategies that are going to be effective to treat different diseases. So try to treat aging or multimorbidities is probably a long game, that when we learn about treating these different diseases with senolytics, we'll be able to move. And at the moment, I think that given the limited number of clinical trials that we have, I think that we still don't know what's necessarily the best strategy. But of course, the ideal strategy, mimicking some of the experiments that we see in mice, is that we should be able to ideally use these senolytics in relatively middle-aged persons, people, once a year or once every few months, and that will keep the number of senescent cells in bay, and that will have an impact in the longer term. But I think that is very, very long way in the future. So I think that we are still very early on senolytics, and I think we still don't have one senolytic drug that has been approved in the market. So the trials that are more advanced are phase 2, and there are a very limited number of trials, so still this far away in the future.
Wonderful. Thank you so much, Jesus. Thank you everyone for your questions. Just give our appreciation to Jesus again. Thank you.